Please.
Okay, good morning to everyone. I'm very pleased to be here to present our invention related to the development of innovative, affordable medicine for the treatment of obesity, diabetes, and neurodegenerative diseases. We made part of this discovery under design of Tuscany Health Ecosystem, particular this book too, and the drug design part of this book. And in this framework, we wanted to realize a new small orally active drug, new small molecules being orally active to treat diabetes and neurodegenerative diseases that are often associated with diabetes. I use this term diabetes because it is amply known and widely accepted that there is a strict association between type 2 diabetes and obesity, pathologies that are often correlated. And in this speech, I wish to communicate to this audience about the possible introduction for the treatment of the diabetes of new orally active small molecules, basically having three major goals that are shown here. So the individuation, the exploitation of a new mechanism of action. Also, an important factor is the reduction of the cost of the medical treatment, otherwise it would be not sustainable for the National Health Service. And the third is an increase in patient compliance by the use of an oral administration. So obesity is a very complex metabolic disease that affects up to 20% of adults worldwide, and it's a very serious factor that affects our health, predisposing our body to several pathologies. So there is not magic pill to cure obesity. However, there are new kind of medicine that are absolute game changers. You have certainly heard about this kind of medicine, this kind of drug, particularly semaglutide and tirzepatide, that are very effective for treating first diabetes and also obesity. These drugs are peptides, very effective peptides, are very famous. But they present also several drawbacks. For example, the very high cost of the medical treatment. The administration should be only by injection, normally, I mean. And also gastrointestinal side effects are very important, in particular nausea and vomiting. Also, a remarkable substantial muscle loss has been observed recently. Moreover, these compounds, being peptides, are not suitable for the treatment of neurodegenerative disease because are not easily permeant to the BBB. So, GLP-1 is an incretin hormone naturally secreted by our body, our gut, I would say, after a meal, and it is known to have several clinical benefits, such as the reduction of glycemic control, so the control of the glycemic, and also neuroprotection and cardioprotection are important effects. Other important effects, especially for the weight management, are the increase in energy expenditure and reduction of appetite. We came into this field in 2018 with a patent disclosing a completely new chemical entity that seems to have the right physicochemical feature to become an oral drug candidate to fight diabetes and obesity. And in general, as said before, the small molecules are easier and cheaper to produce with respect to peptides. And in particular, our compounds can be made in only 4 linear steps for commercial precursor. So are relatively easy to prepare, and the synthesis is scalable. So this is a very important point. Another differentiation of our compounds from existing solution is the fact that these compounds are not GLP-1 receptor agonists, but they act with a different mechanism of action. Just a remark, there is not to date a small molecule drug acting on GLP-1 pathway in the market, and there is a stiff competition for their development. So just having secured the chemical scaffold, you can see the Marcus here. We made a so-called lead optimization, finding lead compounds. You can see the huge difference of activity with respect to the IT previously found. And also in this case, we made other patents securing the intellectual property of our discovery. Just to make to show you the main features of these compounds, we have some proof of concept in vitro about the GLP-1 insulin secretion. You can see the increase in GLP-1 secretion on SCT1 cells, an increase in insulin secretion in INS1 cells, also human cells. This is importantly glucose dependent, the secretion of insulin. We have also an increase of cell viability and mitochondrial activity. This is also a very important point. We have also observed the insulin-like effects on this particular kind of cell. They are of neuroblastoma, human neuroblastoma cell line. And we have also no toxicity on hepatic cell line. About the neuroprotection, anti-inflammatory activity, we have a remarkable neuroprotective action of a neuroblastoma cell line with the substantial increase in 2 major cell survival pathways such as PAKT and MAPK, and a decrease in caspase 3 that on the other hand is involved in neuronal death and apoptosis. And importantly, these effects are superior to liraglutide, that is one of the peptides commercially available that are permeant to the BBB. We have also a marked reduction of the pro-inflammatory interleukin-6 and an increase of anti-inflammatory interleukin-10. Just to show you some experimental details about this, you can see here the prevention of inflammation using several stimuli on human microglial cell line. You can see the effects of our lead compounds. And human microglial cell line are very important because they're the first line of immune defense in our central nervous system. Moreover, in addition to this, they are also involved in other cerebral processes, such as the modulation of the synaptic architecture and also in neurogenesis. So these are very important cells. We have also some proof in vitro on this cell showing the reduction of inflammation induced by amyloid beta proteins that are very important for the Alzheimer development. These proteins are known to accumulate to form amyloid plaques and to cause neurodegeneration and neuroinflammation, as shown here. And we have also optimal in silico drug-like properties. The compounds are permeant to BBB a gastrointestinal membrane. We have no cytochrome inhibition and no genotoxicity. Clearly, we have also to demonstrate this experimentally. It's not just enough to make this in silico. We have also recently some in vivo data about an increased GLP-1 and insulin secretion, glucose-dependent insulin secretion on diabetic mice. And just the last result, because we have also interesting result in chronico with in diabetic mice, giving the drug orally, clearly, because this is a very important point. We observed a significant weight reduction compared to the untreated group, to the positive control, where we used metformin. Okay, so we have a consistent reduction of the weight, and also a significant reduction of triglyceride and glycated hemoglobin. No apparent toxicity was found. So at the end, so just to sum up this talk now. We would like to enter to the clinical trial, but you know that we need a lot of preclinical data to acquire, so we need a complete preclinical package, and to do that we are open to collaboration or to out-licensing our intellectual properties. At the end, I would like to thank all the people of the University of Pisa involved in this project. And clearly the financial support of Spoke 2 of the Tuscany Health Ecosystem is acknowledged. I also would like to acknowledge the University of Pisa for financial support derived from Dimostratori Tecnologici 2022. And these are my contact details. I would like to also to thank you for your kind attention. Thank you. If you have questions, don't hesitate to ask me.
Thank you. Just one quick question. Do we have? Yeah.
So thanks, Mauro, for the presentation. I have just a curiosity. I don't know if I skipped this part, but how did you arrive to the discovery of these molecules? Did you arrive to this by chance or by using virtual screening or what other methodologies?
Thank you for the question, Elena, because this is a normal question that everybody make to me. Because actually I am an organic chemist, and I found just this kind of scaffold just for casualty, I mean, for serendipity, because just sending these kind of molecules, I mean the EAT compounds, to an open innovation platform that was made by Eli Lilly in 8 years ago, around. And just by phenotype screening, we discover the activity. We have not target in mind, but just have this lucky event. Okay, thank you for the question.